Now we are going to see that how the seed germination take place. So first of all, we should know that what is seed germination. Seed germination is actually a period when a dormant embryo just grows and give rise to a new plant. So it is a process when the dormant embryo inside, which is inside the seed, we have already discussed this one, it grows into an active plant. So how? Seed absorb water, it swells up, as a result its seed coat soften and it just burst out. So this is how the seed germination occur, and and we know that plumule is going to form a shoot and radicle is going to form a root. So I think you got it, that they absorb water, they swell, this coat becomes softened and this just burst open, and this give rise to a shoot and this give rise to a root. So this is how the germination occur. Now the germination is different in case of monocot, that is which have one seed leaf, that is one cotyledon, and the one which have two cotyledons. So this kind of germination is called as apigal or hypogal germination. This kind of germination is seen in dicots and hypogal is seen in monocots. So what is there in the apigal? In this, the cotyledons actually, as you can see, that this is the apical one. So what actually happens in this case? This is in cotyledon, right? So from the cotyledon we see that the plant arises. This is inside the soil, even this is inside the soil. It forms a root and the shoot later transforms into this kind of structure, and when we see that the cotyledons are actually above the soil. So that means once first the cotyledon in the soil, they give rise to a small plant, the plumule and radicle, they form root and shoot, and finally in this the cotyledons form the leaves. And the plumule forms the leaves. So what happens in this case? Cotyledons, they are pushed above the ground, they become green firstly. Cotyledons, they are green, they start photosynthesizing, and when they start photosynthesizing and after that they just finally fall off. That means they finally, the photosynthesis stop in them, and then the plumule forms the true leaves of the plant. So this is seen in the dicot and it is called as apigal germination. But in the hypogal, what happened? Cotyledons lie inside the soil only. Suppose this second stage occur, it is occurring in the soil, and then also this is present in the soil only. It gives rise to a plant, and cotyledons remain inside the soil. They have the food stored in them, and that food is actually used up for the growth of the plant, and they don't fall off like that. So they just like the food which is stored in them they are supplying continuously to the plant, which help in the germination. It is seen in the monocot plant. So this is two kind of germination. So if you get a question like this, you are going to write in this that cotyledons, they are pushed up, they become green and they start photosynthesizing, and finally they fall off. And plumule forms the true leaves and radicle form the true roots. But in the hypogal, it is seen in monocot. The cotyledons are going to remain under the ground in the soil, and the food present in them is being used for the growth of the plant. So this is what, and you need to draw the diagrams as well. I have drawn both the diagrams from the same figure, but what you do, you just make these stages for the dicot differently and for the monocot differently for the better understanding of the examiner. So this is all about the seed, the structure, the germination and the type of germination which we observe in monocot and the dicot.